So for exam 2 review, it's going to cover the skeletal, joints, muscles, and nervous. I'm just going to talk about the logistics first before we move into the rest of the materials. So same thing as exam 1, right? So you'll need the Respondus Lockdown browser. Make sure you have an appropriate webcam. For those of you who I contacted for the last one, make sure you have it for this time because, the first time was just to let you know, but the second time I will have to follow through with the consequences of not having the webcam. And if you just, are unsure what is expected from the webcam, I do include a link of the video of someone doing a environmental scan. Environmental scan. Okay, so make sure Response Lockdown browser is on. You can't take it without it anyways, and then have the webcam on. But more importantly, give me a thorough environmental scan. Okay. Then for a lecture, it's going to cover the following topics that I just talked about. Amongst the topics, there are 50 questions. 40 of them are multiple choice. 10 are fill in the blanks. Same as the last one, you're going to see the 40 multiple choice first, followed by the 10 fill in the blanks. It is divided evenly amongst the units. Now, muscle actually has 2 chapters, and nervous has 3. So, because there's more chapters within muscles and nervous, there's going to be a few more questions there. And skeletal, there's only 1 chapter, and joints, there's only 1. So that's kind of where you'll see the distribution of the questions based on how many chapters fall under each topic. You will have 50 minutes to take the exam. And the class average from the last exam is about 36 minutes. So your class clearly, take your time, read the questions slowly, read the questions carefully before you move on to take the next question. Any questions on the lecture exam? Okay, so it is available from Sunday at midnight to Monday at 11 p.m. Central Standard Time. And the Sunday midnight, think of it as like Saturday at 11:59 p.m. Okay. Then for the lab, it's 30 questions. And they're all identification. Spelling counts. Not so much on this one as the next exam, lab exam, but if there are a pair of something, make sure you include right or left. If there's a pair, what else? Write out the full names. Like this one right here. Like kind of several issues. All right. Our point is not.
I think we lost you, Professor, for a second. You're muted.
that was really odd. I don't know what happened, but I got kicked out of Zoom. My PowerPoint froze. Let's try this
Okay.
again. It's still recording though, so that's good. Let me share my screen again. Okay, can you see my PowerPoint?
Yes.
Yes.
Okay. So for example, if it's asking for this bone right here, you know, that would be what?
Distal,
Distal what?
Planix,
Mm-hmm. And 1. So that's what I meant by writing out the full name.
and Roman numbers, right? You want it?
Yep. Yep. So distal phalanx 1. Okay. All right. So for this one, you will have 45 minutes to complete, and spelling counts I already talked about. It is also available from Sunday to Monday from 11 p.m. until Monday 11 p.m. Central Standard Time. Okay. If anything happens, text me. Always give yourself some time. So don't wait until 10 p.m. Monday to take the exam. Okay, that's never, doesn't ever bode well. But you can take them separately. Like you can take one on one day and the other on another day. It doesn't have to be sequential, and it doesn't have to be lecture first, then lab. You can take the lab first, then the lecture. Okay, whatever is more comfortable for you. Okay, any questions about the logistics of the exam?
No.
So for skeletal, you want to make sure you know the difference between what's a short bone, a flat bone, and a regular bone, and a long bone. So I can go, which of the following bone is where its height is equal to its width? What would you say?
Short bone.
Yep. Or I can say the zygomatic is an example of what type of bone?
Irregular.
There you go. So I can use pure definition, or I can use examples, and you would need to know which of the following terms fall into it. Then you also want to know the functions of bones. So it includes movement, which is associated with muscles, because bone can't move by themselves. It has to be in association with muscles. It functions in stabilization, and how it does that is that it acts as a framework for like the organs to settle in, and then for the skin to hang on to. Okay, so what are some so essentially your skeleton forms a framework for the rest of your body. It also functions in protection. How does your bones protect you? Well, it forms cages, right? Like what? What are some examples of cages that your bones form for protection?
Rib
Your
cage,
rib.
Cage.
Exactly. What else?
skull,
Yes. And
Is it the thoracic cavity?
your pelvic. Yep. Okay. So bone also functions in red blood cell production. So if there's a true or false statement, blood cells are produced in the bone marrow, specifically in the bone, that would be true. Okay. And the skeletal system also functions in mineral storage. And what primary mineral is stored?
Calcium.
Yes, thank you. Calcium phosphate. Okay. All right. Now, of the 4 types of bones, short, flat, irregular, and long, I really do focus on the long bone. So a good idea, what you can do is just to draw out a long bone. And label. Okay, so for example, there's this structure. So what would A be?
The diaphyses, or no, sorry, like your medullary cavity.
That's exactly it. Medullary cavity. What's the significance of the medullary cavity?
It's
It's
the
where your
marrow
bone marrow
cavity.
is.
Yeah. So it's a space where you can find bone marrow. Okay, now the bone marrow, the type of bone marrow that you can find in the medullary cavity is going to differ depending on your lifespan. Okay, so if you're younger, it starts off as red bone marrow. As you age, it becomes yellow bone marrow. And then as you become even older, it can become gelatinous bone marrow. So it doesn't necessarily always have red bone marrow in the medullary cavity. Luckily, you do have another place where you also find bone marrow. And where is that? Where else would you find bone marrow?
Would you find it in your heart?
Nope. You find it in the ends of a long bone, specifically the trabecula. Okay, so in the trabecula, you will also find red bone marrow. Okay, and the trabeculas are found, they're the functional units of the ends of a long bone. What do we call these? Or the ends of a long bone called?
Epiphyses.
Yep, epiphysis. So these are the ends of a long bone. And if you want to be detailed, you can say, this is where you would find spongy bone tissue. And in the spongy bone tissue, that's where you'll find the trabeculae. And the trabeculae is where you find the red bone marrow. Okay. Then this area right here, the shaft of a long bone, what do we call that?
Diaphysis.
Perfect. Diaphysis, shaft of, oh, that's an F by the way, shaft of long bone. Okay. Then around the epiphysis, you'll find this cartilaginous material right here. And what do we call this cartilaginous material?
Is that the articular cartilage?
Yes, ma'am. D is the articular cartilage. And the articular cartilage is made up of what?
Was it Highland?
Yes, it is. Then the shaft of a long bone is also covered by this connective tissue. What's E? It goes around bone. So what's the word for around and bone?
And
The periosteum.
Periosteum, very good.
There you go.
Okay. Now the periosteum is kind of like the one on the outside, but there's also a layer on the inside. A, B, C, D, E, F. So what would you call the one on the inside?
that one is the
The
endoperium.
Yep, the endosteum. Okay, so the periosteum contains connective tissue, and that connective tissue actually will, slowly become the tendons that then bind to muscle. Okay, that's kind of like an attachment place for muscles. Now the endosteum is really unique. The endosteum is important because it contains osteogenic cells. So when your bone breaks, it actually is going to heal from the outside inward. Okay, and that's because the inner layer of that connective tissue consists of endosteum, and the endosteum contains osteogenic cells. So if you, as an adult, if you were to break this bone, you would heal from the outside where the endosteum is right here, and it's going to heal inwards. Why? Because of the osteogenic cells. Okay. Now, in between the epiphysis and the diaphysis, you have this area right here. What do we call that? A, B, C, D, E, F, G.
Epiphyseal line, I think you call it.
Yes, it is. Epiphyseal plates. Epiphyseal plates. They actually are also called the metaphysis. Oops, metaphysis. And a common name is the growth plate.
The epiphyseal plate and the epiphyseal line is two different things.
Same. Epiphyseal plate is also known as the metaphysis, or else otherwise the growth plate. Same thing. Different names, same things. So the epiphyseal plates or the metaphysis or the growth plates, what do they contain?
Red marrow.
Nope.
Yellow marrow.
Nope.
You guys have quarters?
Yes. Actually. They contain hyaline cartilage that will transition and become bone tissue during puberty. And that's how you elongate. So if you were to look at a skeleton of a fetus, you would find lots of this epiphyseal plate or metaphysis. But in an adult, it would be just kind of like a thin line. It no longer exists because it transitions from hyaline cartilage to bone tissue, and that's what allows you to grow taller, your limbs to elongate. Okay, so that's pretty much it of the gross anatomy of a long bone. Now, if I cut this long bone in half, like this, okay, here's what you would see. Oops, did not mean to use that color. Let's use this color. Let's use this right here. So this right here is, let's go with letter E and F. So what is that?
She didn't like it as much as before. I think it's just a lot.
So what's EF?
Hey.
What's EF?
Is that the peristem?
Yep.
Okay.
So that's the periosteum and osteum, right? So what does that make this one right here?
That's the osteon, or no, that would be your medullary cavity.
Perfect. Okay, so you need to know what you're looking at for it to make sense. So this is a cut of a long bone. So remember, here's my cut right here. So this is what you're seeing looking down once I cut it in half. Okay, so once I cut it in half and you're looking at down on it, the space right here between the periosteum and the medullary cavity, it's actually composed of these smaller circular tubes. These smaller circular tube, and it's completely packed with them.
Jaden, what do you want for your birthday? A what?
Okay, what are these things? So I'm going to pull one out just to show you what it is. So what is one of these things called?
Is that the osteon?
Osteon.
Yeah, exactly. So that's where an osteon is. So an osteon is found along the diaphysis of the long bone. Okay, so the if I cut a long bone in half, this is what you would see. and I'm going to pull an osteon out. So now what you're looking right here is an osteon, even though it looks awfully like the cut of a long bone. It's not. This now is an osteon. And what is so confusing about is that osteon looks like what you just saw earlier, but without the green lining. Okay, because this is zoomed in on one of these circles right here. So that's what an osteon is. Okay. An osteon also has a center, but now the center is called what?
The central canal.
That's right. What's the significance of the central canal?
It's full of blood vessels and nerves, no?
Yes, ma'am. Contains blood vessels and nerves. Okay, which is why, by the way, breaking a bone, very painful, lots of nerves. Okay, but it does have a blood supply, blood vessels, which means that it will heal. Just takes time and very painful. Okay, now an osteon has these little pockets. These hollow pockets. What are these hollow pockets called?
Is it lemme or
No, ma'am.
Are these the perforating canals that run perpendicular?
No,
No,
ma'am.
because that would be different.
Correct.
lucine?
Yes.
Yay.
Okay, so that's gonna be number 2. So number 2 are the lacunas. A way I remember is that it sounds like a little lake, like lacunae, lake as in lacunae. It looks like a little lake. It looks like a little pond. So that is a hollow space that contains what?
That's
it
where the
osteocytes?
osteocytes.
Yes, ma'am. That is correct. Osteocytes are cells, right? They're living cells, so they need to have a way to pick up nutrients and get rid of waste. And that's why they're connected to each other. So these thin networks, these lines, these little tunnels or these networks. What are these tunnels or these networks called?
Those are the perforating canals?
No, ma'am.
It's smaller than lamellae.
Can I
These,
I
go ahead,
can't say
finish.
Canalixculi? I'm sorry, I might
Yes,
be
it
saying
is. Your app, that's fine. Okay. So
Who
they're
came
called
up with all these names?
some Italian dude discovered this. That's right. So number 3, these thin lines that connect the lacunas are the canaleculi. It really does sound like the snack, right? The dessert. Canaleculi.
They're
Okay.
cannolis.
Yeah, there you go.
There you
Cannolis.
go.
Thank you. There. So these are networks of channels that connect the lacunas. Okay. All right. Then around all of these, in like these concentric rings, what makes the osteons look like a cut of a tree. These concentric rings right here. What are these concentric rings called?
Is that the
lamellae?
lamellae?
Yes, it is. Okay. So the lamellae are concentric rings of matrix. What's matrix consist of?
1/3 collagen, 2/3 hydroxyapatite.
Oh, wow. You got like to the heart of that question. But can we go broader though? In general, what is it? You
Brown
remember
matter?
that because that's really
Brown-
important.
Ground substance?
Yes. Ground substance. Like she went exactly for where I wanted it to end up, but I'm not quite there yet. So ground substance and
Fibers, right?
yes, ma'am. Okay. So the fibers that you will find in the lamella are specifically what? Because remember, bone is strong. You have three types of fibers. Which is the
So it's
strongest fiber?
cartilage.
No, ma'am. Cartilage is not fibers.
It's calcium and phosphate.
That is also not a fiber.
Mhm.
You have three types of fibers. You have collagen, elastin, and reticular fibers.
What would be collagen, no?
Yes, it is. So collagen is the type of fibers that you would find the matrix of a long bone, or more specifically of an osteon. Ground substance. What ground substance would you find in here?
That would be the hydroxyapatite.
Yep, exactly. So within the ground substance, you have the organic and the inorganic. Okay, so the inorganic is so the matrix is the organic and inorganic. The collagen is the organic. The inorganic is the ground substance, and that's the hydroxyapatite. What's hydroxyapatite?
Crystallized salt.
No. You got the crystallized right, but it's not salt.
Is it crystallized matrix?
Yep, but what's the matrix? What substance?
Phosphate, calcium?
Yes, calcium phosphate. So it's not sodium chloride. That's what salt is, right? So it is crystallized, but it's crystallized calcium phosphate. Does that help for you to place where things are?
Yes, that helps a lot. Thank you.
Yes.
Now, an osteon is a tube, right? Because I just said it's a bunch of tubes lining the diaphysis. Now, here's the central canal. I shouldn't have written the osteon there. There we go. Here's the central canal right here, right? Now they're along the perpendicular to it. There are these perpendicular spaces right here. What is that space called?
The perforating canal.
That's where the perforating canal is. Okay? All right. There was a request for cells. So let's talk about the types of cells. So in terms of cells, you'll find the osteogenic cell. And we talked a little bit about that already, right? Think of genic as in genesis, like in, we hear about the genesis of something. What does genesis mean?
Like they can multiply?
Yeah, the creation, right? Stem cells, right? So osteogenic cells are stem cells that will create more. And you're right, a characteristic of osteogenic cells is that it's highly mitotic. It can produce a lot of itself, okay? What did we learn about it? Where can you find osteogenic cells? Remember, which is why your bone heals from outside in.
And the endosteum
An
stem?
osteum, very good. That's one place where you can find it. Where else can you find it?
Periosteum?
Not periosteum, no.
Oh,
And my note says inner layer of periosteum.
That's the endosteum. So,
Would you find it in bone marrow?
yes. Okay. Oops. Here we go. Okay. So here's the periosteum, right? In the green. And then, so that's the E. And then there's the F, right? So the F is the endosteum. So that's the endosteum is the inner layer of the periosteum. So we kind of distinguish it and say the periosteum technically is the outer one and the inner one is the endosteum, which is why I said no to your first response. So the osteogenic cells is found in the endosteum, but you can also find it in the medullary cavity, okay? And usually that's when you're younger, but as you age, mostly it's found in the endosteum, okay? Then the second type of cell that you have is osteoblast. Think of the B in blast as the B in building. So the osteoblasts come from the osteogenic cell, and that's why you see this arrow right here. Let me change that to red. Right here. So the osteogenic cells are going to become osteoblast. The B in blast is the B in building, okay? So osteoblasts are cells that build bone tissue. How does it do that? How do osteoblasts build bone tissue?
They remove calcium from the bloodstream.
They remove, that's right, okay? So they're going to take the calcium from your bloodstream and they're going to put it into the lamella. And they're going to cause the calcium and the phosphate to crystallize and become the hydroxyapatite. And they're going to, you know, pull the calcium, the collagen and everything, and that's how they build it. But what happens is that the osteoblasts are going to build the calcium, phosphate, and the collagen around itself until it becomes trapped. Let that sink in for a little bit. So when the osteoblast build, it's like it's building a wall around itself. So when it builds a wall around itself, it ends up creating this cavity, the space. And what is that space called?
The Lucanlimae. Sorry.
Lacunae, exactly.
Yeah.
So because it becomes trapped, because it builds it forms the hydroxyapatite around it, right? It no longer can do that because now it's trapped and it can't build it can't like pull any more calcium into it. So at this point, the osteoblast is no longer building cells. So it's the same cell. It's the same cell, but it's no longer building bone. So that's why at this point, it's no longer called osteoblast, but now it's called
Osteocyte.
That's right. And that's why you find osteocytes in lacunas. Why? Because the osteoblast was building the wall around itself and it became trapped. And that's the transition. So osteogenic cells become osteoblasts, osteoblast becomes osteocytes. Okay? Now you have a fourth cell. This fourth cell is not related to these three at all. It comes from a completely different, source. Then you have the osteoclast. The osteoclasts are the bone dissolving cells. Now look here. Do you see the osteoclast has these ruffled borders right here? These ruffled borders come by and break down the calcium phosphate. And when it does that, it releases the calcium back to where?
To the bloodstream.
Yes, ma'am. Very good. Okay, so it's got these ruffled borders with the enzyme in it that helps to break down bone tissue. So we always think that like a bone is very stable and it always stays that way, but that's not true. The bone is actually undergoing lots of homeostasis. Whenever you have too much calcium in blood, it takes the blood and stores it in bone. Whenever you don't have enough calcium in blood, it's going to take the calcium from your bone into blood, which is why if you don't have enough calcium in your diet, what happens to your bone?
They get weak.
It get weak. It get very porous, right? Which is a condition called what? That tends to be very common in elderly people.
Osteoporosis?
Yes, ma'am. That's right. Okay. All right. So hopefully that allows you to make connections with the previous slide with some diseases so that it can solidify the different types of cells. You got the osteogenic, the osteoblast, osteocyte, and then the third separate cell is the osteoclast. Okay. Then you have bone tissue. You have the compact bone tissue and the spongy bone tissue. Now, if all of your bone tissue was made up of compact bone tissue, in other words, if every single bone in your body was all osteons, it would be so heavy you couldn't move. You would not survive very well because your skull would be so heavy, it would settle down on your vertebrae and it will damage your spinal cord. Okay, and that's why it's really important that bone have both compact bone tissue and spongy bone tissue. Now, don't confuse it with its ability to be durable and strong. Both compact and spongy are very strong. Okay, so compact is clearly strong because it's like a whole bunch of sticks together. If I gave you one pencil, can you break that one pencil easily?
Yes.
Yeah, but if I gave you a stack of 10 pencils, can you break that easily?
No.
No. And that's what the diaphysis, right? The diaphysis is composed of compact bone tissue because the osteons completely form a bundle around it. Lots of strength. Okay, so that's why compact is heavy and strong. Spongy is not as heavy, but it is very strong. And here's the reason why. Do you see these trabeculas right here? Do you see the trabecula? The shards? Yeah. So it turns out that the shards of the trabecula fall along the stress line. Okay, so it forms along the stress line. So even though it's thinner, it's positioned so well that it allows you to still bear the weight of your body. So spongy bone tissue, although it is not as strong as compact, it is still very strong because the trabecula, the shards within the spongy bone tissue, is formed perfectly along your stress line. So it provides a lot of strength that way. Okay, so that's bone tissue. Then in terms of bone formation, your bones form in two mechanisms. One, endochondrial, two, intramembranous. Endochondrial ossification. Look at its name. Chondrial means it starts with what substance? Midterm people. Chondrial means what?
Cartilage?
Yes, it is. Very good. Endo means what?
Inside.
Yes. So during fetal development, bones actually form in two steps. So in endochondral ossification, first, it lays down a template of hyaline cartilage. Then that hyaline cartilage from the inside outwards starts transitioning from cartilage to bone tissue. So during fetal development, you form bones inside out. Okay. So then it becomes bone. Okay. That's what endochondral ossification means. It means first you have a cartilage template, then from the inside out, you transition to bone. Hence endochondrial. Endo inside out, chondrial because you start with cartilage as a template. Okay. The second type, oh, and by the way, endochondral ossification often forms long bones. Where intramembranous ossification are more of like the flat bones. And what it's going to do is that it's going to form kind of a sandwich. Instead of using chondral, it uses mesenchyme. And you should know that word, mesenchyme. So mesenchyme is the starting material for intramembranous ossification. And what it does is it forms kind of like a sandwich, which then builds flat bones, flat and other types of bones. So those are the two ways in which bone is formed. Okay. So here's another. So I just took a picture of the whiteboard that I use when I teach my in-person class, and I kind of went through with you what I went through with them. Okay. So here's another a nicer drawing, I would say, which is why I included in here. And I purposely didn't label it on the images because I want you to quiz yourself. The answers are written below. So you can go and say what's A, what's B, what's F, what's E, and so on. Okay. I think that's it. that's it for both. Questions before I move on?
Can you go back to slide number 3 just so I can take a picture of it and draw it out again?
I will give you these PowerPoints actually.
Thank you.
Yeah, take a picture. Yep. Sometimes it's better in the moment to have it. Let me know when you're done.
Done. Thank you.
Okay. Faster than I could even take a drink. Take a look at this. Tell me what it's about.
The joints, the type of
Yep.
joint.
What is this? What are you seeing? Oh, actually, it says it on there. It's a cognitive map, right? I highly recommend you going through joints using a cognitive map. Why? This drove me crazy when I was taking this class because all the words sound the same. Synchondrosis, synarthrosis, symphysis. You get what I'm saying? They all look the same, right? So for
Yes.
me, it was really helpful to be able to see how they are organized, okay? So I want you to go, not go home, you're probably already home, but to do this, finish this cognitive map out. I already did the first one for you, okay? So type, fibrous, structure, it's made up of fibers. The type of movement, synarthrosis. So whenever you hear the term synarthrosis, is it a type of joint? Yes or no?
No, it's the
No.
movement.
You two just made my day. Exactly. It is not a type of joint. It's the word for a movement. On the test, I ask students all the time, what is the joint called? And they will give me the term synarthrosis. I'm like, no, synarthrosis is not a joint. It's describing the type of movement. Synarthrosis, amphiarthrosis, diarthrosis, all of those three terms are not types of joints. They're types of movements, right?
Mm-hmm.
Right? Yeah?
Yes.
Okay. So if I ask for a type of joint, I'm expecting things like syndesmosis, synchondrosis, all the other syns, but not synarthrosis. Why? Because synarthrosis is
A movement.
exactly. All right. So I would like you to fill the rest out for me. So you got fibrous joints. What other joints do you have?
Synovial. Ooh.
Oh, you just killed me a little bit. Synovial is a term for what?
Structure.
Yes. So synovial is one. Yep. And what kind of movement does synovial have?
It's free, no?
Huh?
It's free movement.
What's the term for that?
Multi-axial, like ball and joint.
That's the name of the joint axis, but I want to know what the name of the movement is.
Diathesis.
There you go. You see how easy it is to make a mistake there? So you got to make sure you practice where you're using the correct terms. So it's diarthrosis. Okay? So then you would fill the rest out. So there's fibrous, they're cartilaginous, and then there's the bony joints, and then the synovial joints. Okay? So fill this out, and then fill out all the types of diarthrosis. There's the ball and socket, there's the hinge, so on. Okay? All right.
Okay.
I am running out of time. I have 10 minutes left, so I'm going to quickly go through this so that I still have some time to give you some. Actually, you're going to get practice questions in Emma's session. So can I finish the joints up here instead of going through my quiz questions? Or would you prefer to do quiz questions?
I prefer quiz questions personally, but there's,
Yeah.
I'm
We
not the
can do
only
that.
one in class.
We can do that. That's fine. That's completely fine. So I'm just going to quickly go through the rest of my joint slides, okay? So with joints, you also want to make sure you know the specifics of synovial joints. What's the meniscus? What's the bursa? What's the synovial capsule versus the synovial cavity versus the articular cartilage, okay? Define all these for me and be able to identify them. So for the muscles chapters, there's 2 of them. 2 chapters within the muscles. Make sure you know the function, structure. Let me admit students really quick, 1 second. Okay. So from the muscles unit, you want to make sure that you understand the function and structure of the muscles, and then focus on the neuromuscular junction as well as the sliding filament theory. And that's the part where we talk about the four main steps: the excitation, contraction, contraction, and relaxation. And then the metabolism portion of it. So that's the four main things within the muscle lectures: function and structure, neuromuscular junction, which connects directly to the sliding filament theory, which includes excitation, contraction, and relaxation. And then you have the metabolism, and that's where energy source and energy and metabolism occur. The lecture for those two we're in. So to start off, we have the basic kind of the fundamental information about the muscles. So we know that muscles are attached to bone through a tendon. Who remembers what this is? What's the green? What allows for attachment on the bone? So the green portion on the bone that allows for attachment to tendon.
Ligament.
Nope. Ligaments will attach
Ostium.
bone.
Fascia?
The what? I'm sorry, I didn't catch that. Can you say it again?
I said the paramecium.
The paramecium is a fascia that surrounds the fascicles. So
Is it
this,
the little cartilage?
sorry, let me, so on the bone, you see
Epimysium?
this portion right here?
Epimysium?
So I heard a few different things. So I heard about the epimysium, I heard about the paramycium, neither of those. So this green right here is surrounding the long bone, the diaphysis of the long bone, as a matter of fact.
Periosteum.
Periosteum, very good. So that's the periosteum. And that's what I just wanted to talk about it since we just finished talking about it earlier at the earlier session. So that's the periosteum that surrounds the diaphysis of a long bone. And one of its kind of function is to allow for attachment of the tendon. And that's what you see here. So the tendon's here, followed by the muscle, or what we call the muscle group. And the muscle group, just think of like a chicken thigh. You know, you can peel it apart and it comes out really stringy, and that's because the muscle itself is composed of fascicles, and fascicles are bundles that are formed by muscle cell or muscle fiber. Okay, now each of these three kind of divisions have a fascicle. I'm sorry, has a fascia that surrounds them. What's a fascia?
It's a connective tissue.
Yep. Okay. So the fascia or the connective tissue that surrounds the muscle or the muscle group is called the epimysium. Epi meaning surface. And then the paramysium is what surrounds the fascicles. And then you have the endo meaning inside, endomysium, which surrounds the muscle fiber or the muscle cell. Okay. Questions so far about this division? So if I were to take the muscle fiber or the muscle cell and kind of zoom in, this is what it would look like right here. Okay. So you can see that the muscle fiber, that's its technical name, is an actual cell though. Let me let that person in. Check the chat. Okay. So The muscle fiber, which is essentially a muscle cell, has all the components that you would find in a muscle cell, right? Or in just a traditional cell. So it has the plasma membrane, but here the plasma membrane is referred to as the sarcolemma. So some terminology differences. You've got the sarcolemma, and then the cytoplasm is called the sarcoplasm. What's the smooth ER called?
Sarcoplasmic reticulum.
Very good. Sarcoplasmic reticulum. Okay, and then that's pretty much in terms of the terminology that is the major differences. You'll find that a muscle fiber, aka the cell, has a lot of these following structures. So you're going to have chains of glucose right here. What are these chains of glucose called?
Glycogen?
Yeah, that's right. Okay. Then you'll have tons of these molecules right here. So you'll have tons of these proteins that carry oxygen. What are they called?
The myoglobin.
Very good. Later on, when we get to blood, you're going to learn that there's also a protein in blood that carries oxygen, but there it's called hemoglobin. But myo meaning muscle, so it's the protein of the muscle that carries oxygen, so it's called myoglobin. Okay. Then you will find lots of these because muscles, besides the brain, uses the most amount of ATP or energy in the body. What organelle am I talking about here?
Mitochondria.
Yep. Okay. So the exact same muscle fiber, aka muscle cell, has, that's one layer of it. Another layer of it, you will find the myofibrils or the myofilaments that form it. Okay. So what you'll find, oh, we forgot something. What's this thing called? The space, not the letter T that I drew in there.
T-tubule.
Yep. These are the transverse or the T tubules. Okay. Oh, by the way, what is this called? The area where there are receptors, the green little Y structures.
The synaptic knob?
It's not the synaptic knob, actually, because the synaptic knob is part of the neuromuscular junction.
Terminal.
What is it? The synaptic cleft. Yep. Go ahead.
Terminal crispinae. I don't know if I'm saying that right.
The terminals. Yep. The terminal cisternae. Yep. Is
Yeah.
actually a part of the smooth ER right here. So that's not it either. So what area contains the receptors, the neurotransmitter receptors along the sarcolemma?
The synaptic cleft.
The synaptic cleft is the space between the neuron and the muscle cell. We call this the motor end plate. Okay, so the motor end plate is a specific area along the sarcolemma that contains receptors for the neurotransmitter, and in this case, it would be acetylcholine. Okay. All right, so those are the main kind of important structures that distinguish a muscle fiber, aka a muscle cell, from other cells. Okay. All right, then if we add another layer to it, you'll be looking at the myofilaments. So in the myofilaments, there are two main myofilaments. What are they? The thick and thin?
Actin and myosin.
Yep. So what would the thin one be called?
Actin.
Yep. And the thin ones are attached to what?
Troponin and tropomyosin.
Yep. So the troponin, tropomyosin are connected to the actin, but the actin within a muscle fiber is attached to what?
The Z disc?
Yep. So these are the Z discs right here, which makes this structure then actin, or otherwise the thin filaments. Okay, that makes this structure right here what?
The myosin?
Yep. And myosin is also known as the
Thick filament.
thick filament.
Filament.
Yep. And myosin is distinguished because it has these things right here. So on myosin, what will you find?
The heads?
Yep. Myosin heads. What's the significance of the myosin heads?
They need energy to like stick up away from the myosin.
Excellent. So they need ATP to be able to kind of cock or bend so that they can bind to the actin. Now, how do they bind to the actin? So here's the purple. So this was actin right here. How do they bind to actin? What must actin have?
The binding site open.
For,
Green.
yep. So these are the binding sites for the myosin heads to grab onto and then pull. But normally, when you're not contracting, what happens? What is it covered by?
The tropomyosin.
Yep. Okay. So it's going to be covered by the tropomyosin troponin. Which is why contraction doesn't happen until you tell it to happen. Okay, so the default is that the muscles are relaxed. They are not contracted because by default, the binding sites on actin are covered by the troponin tropomyosin complexes. Okay. So what's the function of tropomyosin and troponin?
To bind with calcium?
It does do that, but what is their purpose? What's the goal of the troponin tropomyosin?
Muscle contraction.
Does it allow for it or does it prevent it?
It's relaxation, no? Prevents it and then relaxes it.
So if nothing happens, would the troponin tropomyosin allow for contraction or prevent contraction?
Prevent it.
It prevents it. Exactly. So sometimes we use the term TNT system, the troponin tropomyosin system. So the troponin tropomyosin system, the function of that or the purpose of that is to prevent contraction. Why is it preventing contraction? Or how is it preventing contraction?
Because it's not binded to calcium, which doesn't uncover. If it's not bound to calcium, then it doesn't uncover that binding sites.
Exactly. Yep. So let me ask you a question. Would you normally want your muscles to be contracted, or do you want it to be relaxed as a default?
Relax.
Exactly.
Relaxed.
So as a default, your muscles are relaxed. And the reason why they are relaxed is because the troponin and tropomyosin prevent contraction. And how do they prevent contraction? By covering the binding sites on actin. So even if the myosin have energy, have ATP, it can't bind to actin, which is what you want it to happen. You know, you don't want your muscles to be all contracted all the time. You only want it to contract when you tell it to contract, right? And you can control that by releasing what into the muscle cell?
Calcium.
That's right. Because why?
Because calcium is attracted to, tropomyosin is attracted to calcium.
Which one binds to calcium?
The TNT system?
Which part of the TNT system?
The tropomyosin, I think it's what you call it.
Troponin.
Yeah, so it's
Troponin.
the that binds to calcium. Okay, so it's the troponin that binds to calcium. So calcium would bind the troponin, and when it binds to the troponin pull the tropomyosin, and the tropomyosin, which was previously covering the binding sites on actin, shift away from those binding sites, which then allow the myosin heads to grab on and pull and contract. Make sense?
Yes.
Okay, wonderful.
Yes.
Okay. So we kind of went in a little bit about contraction array, which is fine. So it's kind of like layers of an onion, right? You peel a little bit away, a little bit more, a little bit more. Okay. But before we go into the actual sliding filament theory, let's talk a little bit about the sarcomere. Okay. So we talked about the Z disc. Now it turns out that between two Z discs, just that area is not an actual structure, but between two Z discs, you have an area called the sarcomere. And the sarcomere is significant because that's technically like the functional unit, even though it's just like an area and not an actual physical thing. The sarcomere is the functional unit of a muscle cell. And the reason for that is because when we're talking about contraction, it's the sarcomere that shortens when myosin heads pull on actin. That it's that space that becomes shorter. And that's why you have to know what a sarcomere is. So what is a sarcomere? Can someone tell me what it is again?
The distance between the Z.
Yep, the distance.
Functional unit of a muscle.
Perfect. Functional unit of a muscle fiber, aka muscle cell, and it is the space between two Z discs. And the reason why it's the functional unit is because that's the space that decreases whenever contraction happens. Okay, so when you have each of those sarcomeres decrease, the whole muscle fiber shortens. And that, that's what results from the sarcomere shortening. Okay. All right. So that's kind of the basics of a muscle cell organization. So you've got the muscle, which is covered by the epimysium. You've got the fascicles, which is covered by the perimysium. Then you've got the muscle fiber, which is covered by the endomysium. The muscle fiber itself is a cell that consists of the sarcolemma, the sarcoplasm, the sarcoplasmic reticulum, the motor end plate, tons of mitochondria, tons of glycogen, and lots of myoglobin. And if we're looking at myofilaments within that muscle fiber, aka muscle cell, you have two main ones. You've got actin and myosin, and you now know where they're located, what kind of things are associated with them. So troponin and tropomyosin is associated with actin, where myosin heads are associated with the myosin. Okay. All right. So at this point, we can talk about the sliding filament theory. But my question for you is, do you want me to focus on the muscle sliding filament theory more, or do you want me to cover more of the nervous chapters?
I feel like nervous is a little bit more complicated, but I don't know how you all feel about it.
I would like to do the nervous as well.
Agree.
Okay, so let's do. So the sliding filament theory, I already numbered it out for everybody, so you can see from number 1, 2, 3, and you can follow through this this PowerPoint to kind of go through the sliding filament theory. And there's lots of videos that you can watch on it too. So I'm going to keep going and get into the nervous system. And then if I do have time at the end, I'll come back, okay? But most likely I'll be spending the next 20 minutes on the nervous, and then I want to save some time to do quiz questions too, if you like, okay? All right, so the nervous system, let's do a broad overview of the nervous system. So the nervous system is divided into two main kind of divisions. You've got the central nervous system and the peripheral nervous system. Anything nerve related falls under the peripheral nervous system. And of course, you have two types of nerves. You've got sensory nerves and you've got motor nerves. Okay. Under sensory, so oh, by the way, what's the difference between sensory versus motor nerves or within the peripheral nervous system?
Sensory carries sensation to the central nervous system.
Yep. And motor?
And the motor division carries the information from the brain. It's the spinal cord.
Perfect. You nailed it right. Exactly. So the main difference between sensory and motor is the direction in which it carries the information. Sensory will carry the information from the outside into the body. Motor is going to carry information from the central nervous system out. Okay, so both of them are neurons, or bundles of neurons, which is what a nerve is, right? It's the direction in which they carry the information. Okay, so just remember that. The major difference between sensory and motor is the direction. Sensory is going to bring information from the environment internally. Motor is going to take the information internally and send it out. Okay, so within these two, sensory is divided further into what we call somatic versus visceral. What's the difference between somatic versus visceral?
Your somatic, you said it's a skeletal muscles versus visceral is smooth cardiac and glands.
Excellent. Perfect. I couldn't define it any better. So somatic is more of the skeletal, and visceral is the internal organs, the glands, and so on, right? So that's why oftentimes somatic is associated with what? Voluntary or involuntary?
Voluntary.
Perfect. Where visceral is associated with what?
Involuntary.
Excellent. Now you'll notice that both sensory and motor have somatic and visceral subdivisions. Okay? Because again, they're both types of neurons, and it just the only major difference is what direction are they carrying the information. So of course, they're going to have both somatic and visceral divisions. Okay? Now the motor gets a lot more extensive. Okay? So within the motor, in other words, if you're carrying information from the central nervous system out, you do have the visceral and you have the somatic. The somatic is pretty straightforward. It involves the skeletal muscle, like one of your peers just said, and it is voluntary. Where the visceral are things that will go to your internal organs, your glands, and so on, right? Now, the visceral, because it is involuntary, it often goes by the term ANS. What does ANS stand for?
Autonomic nervous system.
Perfect. Autonomic nervous system. Okay. So in other words, this is completely involuntary. You don't have control over this. Okay. Now it is divided further. So the visceral within the motor is divided further into two main responses. You've got the sympathetic autonomic nervous system response, and you've got the parasympathetic response. Now, the sympathetic is like your fight or your fight. You don't have control over it, but when it is activated, it's more of a stimulant. Where the parasympathetic is more of your rest and digest. Okay? Let me give you a scenario. So let's say you're walking down a really dark alley, and all of a sudden a cat jumps across you. What is your reaction?
Fight or flight.
Like,
Perfect. It's your sympathetic nervous system gets activated, and you get, your response is either to fight or to flight. In other words, runner, you're going to stand your ground and you're going to fight, your muscles get tense, or you're going to run. Okay? So that's an example of visceral sympathetic. Okay. Here's another example. Let's say it's Thanksgiving, you know, and some people, not all, but some people have a tradition of eating a lot of food, like turkey, right, during Thanksgiving. And when you eat a lot of food, all your blood, does it go to your muscles or does it go to your stomach?
Stomach.
That's right. So all of that blood rushes to your stomach, and that's because it senses that you're in a safe environment. It's going to take all the blood and put it into your stomach so that you can start to digest. And when that happens, you kind of get tired, you get a little sleepy. You know, just like whenever you eat a really large meal, what happens after your large meal? You get sleepy, right? And that's because the parasympathetic nervous system gets activated. Okay? Do you have questions?
No, that's pretty straightforward. Thank you.
Awesome. Then you have the central nervous system. The central nervous system is going to include your brain and your spinal cord. So that's pretty straightforward. Okay? Then you have, if we were to take a look at a neuron specifically, okay? So if we were to take a look at a neuron, the basic structures of a neuron, you have the dendrites, you have the soma, and then you have the axon. Okay? So the functions of the dendrites are what again?
They send stimuli and send it to the soma?
They receive stimulus. They receive the stimulus, and you're right, they do send it to the soma.
I have a question of that. Right now in the
Yeah.
picture with Emma, she mentioned the motor unit and the motor neurons. So the motor neuron would be the whole thing, right? Including the dendrites, the soma, and the axon?
Yep. So the motor, so you have two types of neurons. You have sensory neuron and motor neuron. Both motor and sensory neuron all have the same three parts: dendrites, soma, and axon.
Got it. Okay.
The motor unit is something else. Okay. So you've got the motor neuron versus the motor unit. Those are two different things.
Okay.
Okay. So a motor neuron, regardless of whether you're looking at a motor neuron or a sensory neuron, they're both neurons. All neurons have these three components: the dendrites, the soma, and the axon.
Okay. Thank you.
Yep. So the dendrites, they have these receptors on them, and that's why they can receive the information. Do you find these receptors anywhere else along the neuron?
No.
No. And that's why information only moves in one direction. Okay, so information only moves in one direction. It's received by the dendrites, moves to the soma, to the axon, and then something else happens. Okay, the information is either passed or lost. Okay, so the dendrites receive information, the soma carries it then to the axon. Now the axon is a little bit more complicated than just the axon. Okay, you've got this portion right here, which is what? 3A is what?
The axon hillock.
Yep, you've got the axon hillock. Yep. And then you've got the axon itself. And then technically, this is a simplified drawing, but technically it branches outwards, right? There's branches. So that will be 3. So 3B is the actual length of the axon. 3C is going to be what?
Is it the terminals?
Terminal arborization. So right now, if I flipped this image, rotate it to the left, it looks like a tree, right? You see the branches and then you see the trunk of a tree, the axon as the trunk of the tree. So that's where it gets the term terminal arborization. Arborization meaning tree, terminal because it's the end. Now at the end of the terminal arborization, you do have this. And what is that called?
Synaptic knob.
That's right.
Synaptic.
That's where the synaptic knob comes into play. Okay. Now, if I zoomed in on a synaptic knob, okay, it has what on the inside?
Vesicles.
Yep. And these vesicles contain what?
Neurotransmitter.
Neurotransmitters.
That's right. Neurotransmitters.
ACH.
Ah, careful. Hold on. Let me finish writing this out. Neurotransmitters. Now, if you're looking, if this motor neuron happened to be next to a muscle fiber, aka muscle cell, then that neurotransmitter specifically is going to be what?
Acetylcholine.
That's right. But there's lots of other neurotransmitters.
Right.
Okay. So if this is the case, we would call this the presynaptic neuron because it's planning to send the information. And in this case, the muscle fiber is the effector, what's receiving it. Okay. But just as likely, rather than having a muscle fiber there, I could just as well have another what?
Neuron.
That's right. In which case, then this one would be a postsynaptic neuron because after the synapse or the transfer of information, this would be the postsynaptic neuron.
Yeah.
Okay. All right. Then looking at the spinal cord, a couple of numbers to know. There's 31 pairs of spinal cord altogether, 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccyx. Technically, the coccyx is several that's fused together, and same as the sacral. It's 5 that's fused together. Okay. A nice way to remember that is you have breakfast at 8, lunch at noon, dinner at 5. And then technically the sacrals are all fused together. Okay. So those are the, oh, sorry, they're not fused. I'm talking about the vertebrae. So you do have 5 pairs of sacral spinal cord, spinal nerves and 1 coccyx nerve. Okay. Now the spinal cord is looks kind of like an oval structure or a butt, then it has kind of a butterfly on the inside. So the butterfly shape on the inside is what we call gray matter, and everything else is the white matter. You should know what the difference between the horns are, the posterior versus the anterior horns. You should know what a posterior root versus an anterior root is. You should know what a posterior root ganglion versus then what happens when it becomes a spinal nerve. Okay. So one way to kind of understand the spinal cord, kind of the way it's shaped the way it is, it makes sense because it allows for the neurons to enter and exit. So in the butterfly area right here, so essentially if you're looking at the anterior horns, usually you'll find the anterior horns bulge a little bit. You see how it's kind of thick right here? Let me use this. So the anterior horns are a little bit thicker compared to the posterior horns, correct? Do you see that?
Yes.
Yes.
And it should make sense because in the anterior horns, you will find the somas. I'm sorry, you'll find the dendrites and the somas of what type of neuron? Is this a motor neuron or a sensory neuron?
Sensory.
Sensory.
Okay, let's review this one more time. So the neuron receives information only in one direction, right? Does it receive information from the dendrites or from the synaptic knob?
Synaptic knob.
Where does it receive the information?
Oh, see the
The
dendrites.
Dendrites, that's right. So this neuron right here, the information is going to be carried this way. Right?
Yes.
So now, is this a motor neuron or a sensory neuron?
Motor.
That's right. And that's what,
Because it's going one way, correct? Okay.
that's correct. Yep. And that also explains why the anterior horns are thicker, because it's got to make room for the dendrites and the soma. Where the posterior horns are smaller, because the soma and dendrites, which are a little bit bigger, are located externally. So like right here. And it's carrying information in this way, which means down here it's just the synaptic knob. So it's smaller. And that's why the posterior horns are thinner. Do you see it right here? They're just thinner overall. Make sense?
Yes.
Yes.
Awesome. So then with the brain, I'm not going to spend too much time on the brain because all of you did a really good job on it. So but you want to know about the major regions of the brain. So you've got the cerebrum, the diencephalon, the brainstem, and the cerebellum. Some of them have subdivisions, like the cerebrum would have the lobes. Make sure you know the lobes and their function. The diencephalon is divided into the thalamus and the hypothalamus. And the brainstem is divided into the three parts. What are the three parts?
Pons.
Midbrain, pons, and medulla.
Medulla oblongata.
Let's start with number one. What's number one?
Midbrain.
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 Brain.
Number two?
Pons.
Pons.
Number three?
Medulla.
Oblongata.
Medulla oblongata.
Medulla oblongata. If you ever watch Waterboy, you will always remember the medulla oblongata because I'm serious. Adam Sandler in the Waterboy, he's like, he's breathing really heavily, right?
Yeah.
So the professor is like,
Yeah.
what's wrong with your medulla oblongata? Why? What's the function of the medulla oblongata?
Respiration.
Yeah, exactly. Okay, now you know. If you watch it again, you'll appreciate it more. All right. So this is probably why a lot of you are here, is to learn about the nerve impulse conduction. Okay. So before we go on to the nerve impulse conduction, there are 4 terms you need to know. The difference between resting membrane potential, local membrane potential, threshold potential, and action potential. All potentials. So what does a potential mean?
The difference of a charge from like
Yeah.
one place to another.
Yep. So we often say that it's the state of the membrane. And in this case, it's the membrane of the neuron. Okay. So here you have dendrites and soma, and then you have the axon and synaptic knob. Okay. So if I were to take a measurement of the electricity that's currently going on in there right now, what measurement would it give me at resting membrane potential?
Negative.
Negative 70 millivolts.
I don't usually make you memorize numbers, but you will want to memorize these numbers. Okay. So this is negative 70 millivolts. So that's easy to remember. Okay. But you have to explain to me why. Now, in order to explain to me why it's negative 70, it's often good to know what is on the outside and what's on the inside. ECF stands for extracellular fluid. ICF stands for intracellular fluid. What's on the outside?
Sodium.
Sodium.
Sodium. Lots and lots of sodium. What's on the inside?
Potassium.
Potassium. Proteins.
Where's the problem here? Why doesn't it make sense from what I've drawn so far?
Because there's a lot of protein in the ICF that make it negative.
That's right.
Yes.
Both of these are positive. So where the heck did the negative 70 come from? And you're absolutely right. On the inside, you have things that don't move around, right? So these are the larger proteins that are negatively charged, but they do not move. That's a negative sign inside with the red negative sign, okay? So that's why it's negative 70 millivolts. So currently, you have potassium on the inside, you have sodium on the outside, but then because of these negatively charged proteins right here, overall, it's still a negative 70. Nothing's happening. Okay, but still, the overall charge of the membrane is negatively charged with sodium on the outside, potassium on the inside, and you have the negative proteins. Questions?
No.
Awesome. Okay. Now, let's say you receive a stimulus. Okay. And the stimulus is going to be a chemical, right? Because on the dendrites, you will find these receptors, which we talked about earlier, right? So let's say the brain sends the stimulus in the form of this chemical. The chemical binds to the receptors on the dendrites, and voila. What color should I use? Let's use yellow. So when that happens, a signal is generated along specifically the dendrites and the soma. Okay, so I'm only looking at this region right here. That's what the local membrane potential is referring to. It's just what's happening at the dendrites and the soma. Okay, so the neuron receives the stimulus and has to decide whether it's strong enough to move along the axon or not. Okay, how many of you have ever ridden a roller coaster?
Yes.
I have.
Yeah?
Yes.
Okay. So when you're going up that roller coaster, you can be like, oh no, I need to get off. And they'll be like, okay, fine. They'll be really annoyed, but they'll let you off. But once you reach that peak,
Polarizing.
yeah, once you're up at the top of that peak, once you go, can you stop?
No.
You're
No.
screwed, right? You're going to pee your pants if it's necessary, but you can't stop.
Yikes.
That's the same thing here. So when the signals are traveling along the dendrites and the soma, it's like slowly going up, building up that climax, right? But once it reaches that threshold or that peak, and once it goes down, you cannot stop it. So that's the difference between a local membrane potential versus an action potential. Local membrane potential, you can still reverse it. And there's gradients. There's some stronger, some weaker. Okay. But once an action potential has been generated, that means like you've gone past the point of return. It's just going to go. Okay. So a local potential only along the dendrites and the soma, right? The stimulus is graded. In other words, it could be stronger or weaker, and it's reversible at this point. Questions? Okay.
The low is when we reach the negative 55, right? Anything?
Yes, that's
Yeah,
what I'm getting to next. Exactly. You got it. Okay. So let's say the signal was strong enough. Let's say the signal was strong enough to reach the beginning of the axon. Remind me what's the beginning of the axon again?
Axon hillock.
Yes, ma'am. Okay. It can only reach the axon hillock if there's a 15 millivolt difference. In other words, if it can cause it to go from negative 70 to what? Negative what? Who's good at math?
Negative 55.
55. That's the 15 millivolt difference. So if the stimulus is strong enough to reach negative 55, a 15 millivolt difference, then you've reached threshold. Where does threshold occur? Where along a neuron does threshold occur?
In the axon
Axon?
The axon what?
hillock.
Hillock.
Hillock. Perfect. Okay. So at the axon hillock, that's when you know you've reached threshold. So that's why oftentimes threshold is also known as, or the axon hillock is known as the point of no return. Or I don't like this term, but it's also referred to as the trigger zone. Because once you pull the trigger, you can't retract it. I don't like that term though. I like the point of no return better. Okay. All right. So let's say the 15 different millivolt difference is achieved. Now the signal is going to be passed along the axon until it reaches the end. Okay. Action potentials are non-graded. They are not reversible. Okay. So that's the major difference between an action potential and a local potential, in addition to where it occurs. A local membrane potential occurs only where?
The soma.
The soma, the dendrites in the soma. Where an action potential occurs where?
The axon.
That's right.
Axon and synaptic knob, right?
Technically, yes. Okay. Now, we're going to take a closer look to the axon. I'm sorry. We're going to take a closer look at the action potential. Action potential actually has two steps. What are the two steps in an action potential?
It's all or nothing.
Well, technically that's the threshold, right? So once it reaches the all or
As in
nothing,
Irreversible.
I'm sorry? Those are the characteristics, but what are the two actions in an action potential?
Depolarize and repolarize.
Yes, ma'am. Depolarization and repolarization. Okay. Look at those terms. Oh, I wrote it wrong. That's repolarizing, not two depolarizations. Now, at resting membrane potential, that's referred to as polarized, right? And polarized is when it's what? Charge.
Negative 70. When it's
Negatively.
neutral.
Negative. It's negative charge. Okay. Now, if it's depolarized, what do you expect it to be? More negative?
Positively charged.
Positive. That's right.
Plus 35.
That's right. And if it's repolarized, it's returning to being polarized. That means it's going to be what again?
Negative again.
That's right. So in an action potential, you have to have a change in the membrane potential. It's got to go from negative to positive back to negative. Okay. Now, you're probably wondering why? Why? Right? Imagine this. If you have a plastic ball on top of water, and if that water doesn't move, does that ball ever move?
No.
No.
It doesn't. What do you need to do in order to cause that plastic ball to move along the water?
Shake it up. Kind of mix it around.
What's another way of saying it?
Move the water.
And when you move the water, what do you create in the water?
Waves.
Waves. You have the wave going up, you have the wave going down, up, down, up, down. And as it moves up and down, the ball also moves up and down. Or sorry, the ball moves along, right? That's the similar thing to an action potential. That's why you have to go from negative to positive to negative. Think of it as waves. Negative to positive to negative, and then negative to positive to negative, then negative, positive to negative, and so on. So this segment of the membrane right here goes from polarized to depolarized to repolarized. The stimulus moves down one area. Then this area becomes negative to positive to negative. Then that moves the stimulus down to the next area. Then the next area goes negative. You get the point, right? I'm not going to say that throughout the whole thing. But that's why you have to have an action potential to move the stimulus along. Does that help?
Yes,
Yes.
that's saltary something. I don't remember.
Yep.
But it's-
So, yep,
Saltatory
we're not
conduction.
going to worry. Yep, we're not going to worry about saltatory conduction quite yet.
okay.
Okay, we're just going to do the basics to understand why you have to understand why you have to know action potential involves depolarization and repolarization. Because when I was learning this, I'm like, why can't it just pass? Why does it have to go depolarize, then repolarize, then polarize? You know, so that's why. To move it along, you have to have that change in membrane potential. Okay, now how do I make it go from negative to positive to negative? What has to happen for depolarization to happen?
Potassium gets out, I think, of the ICO.
No, ma'am. If potassium moves out, it would become even more negative.
Then sodium comes in.
Sodium flows in.
So what? There you go. The first thing that has to happen is the sodium in depolarization. Sodium gates open. And of course, things are going to go from what to what?
Positive to negative.
No, ma'am. Concentration gradient.
It's gonna be
High to low.
High to low. Where is it currently high?
Outside.
On the
easy.
outside.
That's right. So when sodium gates open, sodium is going to enter. Why? It just diffuses in from high to low.
Because they're attracted. The gates separate the attraction.
There's no attraction. It's just a movement, a concentration movement. There's a lot of it outside, so it's going to move to where it's low, which is going to be where?
The inside. On the
On the inside. Okay. So when sodium moves in, now on the inside, in the ICF, you had the potassium, okay, and yeah, you had the negative charge proteins, but now what did you gain?
Sodium.
Positive sodium.
And that's what drives it. Yep. And that's what drives it from negative 70 to what?
To negative 50 to 35. Okay.
35.
Plus 30.
Positive 30.
That's right. Okay. So now that's only going to move the stimulus just a little bit. To fully move it to the next area, you have to undergo repolarization. What's repolarization again? Positive or negative?
Going back to negative.
Going back to negative. How am I going to make this go back to negative? What has to happen?
Sodium gates close.
Yes, ma'am.
And potassium gates open.
potassium opens.
So it closes so sodium can't come in anymore. And that's not completely true, but it's more complicated. So I'm just trying to keep it simple. Sodium gates close so sodium doesn't come in anymore. Potassium gates open. And when potassium gates open, what direction are they going to go and why?
They go out of the cell because they want to go with the concentration gradient.
That's right. Because there's less potassium on the outside than there is on the inside, so they're going to rush out. Okay, so potassium is going to rush out. So now, if I keep track of what's on the inside, now it's sodium, and I've got the negative charge. And that's why it goes from it goes from 35 to what?
Negative 70.
And not only does it go to negative 70, but it's even going to go lower than negative 70.
Negative 90.
Yeah.
Negative 90.
And that's when it goes from an action potential.
To a resting membrane?
No, ma'am. To hyperpolarize.
Hyperpolarization.
Okay.
So make sure you understand what hyperpolarize is. Okay. But For now, we're just going to keep it simple. Resting membrane potential, local membrane potential, threshold, and action potential. Make sense? Questions?
So when it's hyperpolarized, that's the only time it'll be -90.
Mm-hmm.
And when it's just resting potential, it'll be the -70.
Mm-hmm.
Okay, just making sure.
And then
That's a great question, though. 1 second. Let me follow up with her question. Yes. So the reason why I said that's a great question is, so now that once it goes through depolarization, repolarization, gets hyperpolarized, the signal has transferred. So the signal now has moved on, and then that repeats again here, and then that repeats again here, here, until it reaches the synaptic knob. When it reaches the synaptic knob, it causes the calcium gates to open, and calcium enters. And when calcium enters, the synaptic vesicles push out acetylcholine or some other neurotransmitters, right? And then that passes through this space, binds to wherever the receptors are for the next effector, and then that's how the synapse happens and the signal gets carried on. All right, so imagine that's done. Now, if another signal wants to be sent, wants to be carried along this exact same neuron, and it's the same strength, will it be carried?
No,
it has to be.
No. Why?
because it has. Well, if it's in the refractory period, it has to be stronger.
Excellent. Because what happens is now you have sodium inside and you have potassium outside. Is that the same as resting membrane potential?
No.
No. They're both negative, but it's not the same. So in order for it to go from hyperpolarized back to being polarized, you actually have to take the potassium, pump it back in, and the sodium and pump it out so that you can go back exactly to where resting membrane potential is. Okay, unless, what's the exception so that this exact same neuron can carry a secondary signal?
It has to be stronger than the first one.
You got it. Perfect. Yep. All right, questions? Okay, so that's the same thing here. Okay, you can see resting membrane potential, you can see action potential, and then you have to have what's called a charge restoration. You have to take the potassium back in, the sodium back out, and then it can go back to resting membrane potential. Okay. So I like this one because this one went through what we just talked about, but not only does it have the electrophysiology of the resting membrane potential, the local potential, threshold, action potential, but it also goes hand in hand with the graph. Okay, so what it actually looks like. So A right here, that's A here. B right here is what?
The local potential.
Yep. And D right here?
Polarization.
E right here?
Repolarization.
Polarization.
F right here?
Hyperpolarization.
Yep,
Polarization.
that's right. And then A goes back to resting membrane potential. Okay.